couple them together and build a whole universe of interacting fields, just like the standard model. There's one thing I want to do a little bit differently this time around, though. I want to motivate it differently. So in the first part of the talk, I've spent most of the time with the basics, how to treat these kinds of mathematical structures, how to get an analytical insight and the numerical insights. Now I want to present the kind of work how I would present it to a more biological audience. Because one of the key challenges that we face every day is to communicate math-heavy work so that people from a different scientific culture and different scholarly language can find it exciting and useful. And I thought it might be useful for a summer school to do that in the second part of the talk, now that you know how this stuff actually works. I'll add a little bit more math in the talk than I would usually do, but the motivation is going to be exactly how I would motivate this to a neuroscience audience. Okay, couple of maps. Right, so let's go back to the data. You have to do this first. The first study that measured pinwheel densities reliably in 3 species, tree shrew, galago, and ferret, was done by Matthias Kaschuber and Fred Wolf in 2010. Here, this plot shows for a relatively large number of animals. Every symbol is an animal. The size of the symbol is the size of the imaging area of that particular sample. And on the 2 axes, you have the size of the hypercolumn. So that's this lambda squared, the typical scale of the animal. You can see the tree shrew has relatively large orientation domains, relatively small orientation domains, and the ferret has relatively large orientation domains. And here you see pinwheels in their physical unit. How many of these dots per square millimeter? But then we can normalize out the spatial scale using these wavelet-based tools. And then it turns out that the grand average across all of these species is super, super close to 3.14. And when they published this paper in Science, there's a fantastic commentary by Ken Miller, who titled it with that title, which I thought is very clickbaity, but also very, very effective, because that's how the visual system is organized. Okay, since then we have added a few more species, most importantly cat and macaque. Here's the same kind of plot, this time plotted against body weight. So here you see every single animal, the pinwheel density per square millimeter as a function of the body weight of that specific animal that underwent an imaging experiment, and color-coded are the different species. If we measure the column spacing, again, we are all over the place. The macaques are relatively far out because they are so heavy. Macaque and cats are the heaviest creatures here, and tree shrews are the lightest. And if we now compute the pinwheel density in these intrinsic units, we again find that we are super, super close to 3.14 as the ensemble average. Okay, if we look at the evolutionary relationship between these organisms, ferrets and cats are relatively closely related in the group Carnivora, whereas the primates, galago, macaque, ourselves, are also very closely related. And then a little bit further off is the tree shrew. Interestingly, we talked about the salt and pepper pattern previously. We are more closely related to mice and rabbits, for that matter, than we are with cats. And our likely ancestor, early here, just after the extinction event that killed off the dinosaurs, there was a very small, likely dark-dwelling creature with a tiny brain, at least judging from the fossil record. So it's very difficult to picture this organism to have a complicated layout of orientation domains in their brains, just because the brains were so small. And if that is true, then orientation domains have evolved at least twice in the lineage of the mammals, among the carnivores and the primates. And quite remarkably, they have done this with statistical properties that are essentially indistinguishable. And that smells like convergent evolution. Now, if that is true, one wonders how much one can challenge these circuits that give rise to orientation selectivity, and whether one can find even deviations from this common design. If we want to test this hypothesis, we need a group of animals where we have a relatively clear understanding of their evolutionary relationships. They have to be highly visual, and they should have properties in addition to orientation selectivity that might make it difficult for these visual systems to fulfill all of these different criteria at the same time. Now, there is one group of animals that is particularly good for questions like this. And these are the primates. So here are a few examples that will end up in our later data set. The left two are two types of macaque monkeys. Then there's the galago and the owl monkey. Primates have had a fantastic run over the last few tens of millions of years, last 30 to 40 million years. They have diversified in many different ecomorphotypes. The two macaques here, one is jungle dwelling and one is one of those Japanese macaques who likes to sit in hot tubs. The galago is a dark dwelling creature, the owl monkey too. Their color vision is all over the place. Some of the macaque monkeys can see 3 colors. They have 3 cone receptors in their retinas. The owl monkey and the galago are colorblind. They only have 1 cone receptor left that is processed by their brains. Among the New World primates, there are species where only the females are trichromates because the cone information is carried on the X chromosome and most of the males are colorblind. And so there's a whole zoo of unique challenges that all of these visual systems had to address. So if we can collect a data set of orientation maps from these different primates, we might find something interesting there that is actually different between them. Primates also have this property that there is one additional map beyond orientation selectivity that's relatively easy to measure that has remarkable properties. And these are called cytochrome oxidase. That is a particular staining you can do. So you take out the brain, you slice it in thin slices and stain for cytochrome oxidase. And you find these dark regions. These are called blobs. And in this paper, this is data from Margaret Livingston, they measured orientation selectivity, shown here as these radial plots, along an electrode penetration experiment. And long story short, whenever you are in these brighter regions without much cytochrome oxidase, you have larger firing rates for particular orientations. So the neurons are well tuned. But whenever you are in a blob, tuning becomes worse. And that suggests that there's an interesting interaction here between this real valued field, cytochrome oxidase strength, and the orientation selectivity that's part of our complex field that we had discussed so far. And what makes this interesting is that, well, it exists in many different primates, and it's relatively easy to measure. We can measure orientation selectivity through intrinsic signal imaging, and then in the same parts of the brain, we can stain for cytochrome oxidase and get these real valued fields that now live on a grayscale. And yeah, the little squares show magnifications of what this looks like if you were to zoom in. In this field, there was a long-standing observation that there seems to be a weak anticorrelation between orientation selectivity and cytochrome oxidase expression, just like in Margaret Livingston's experiment here. But this anticorrelation is relatively weak. Sometimes you can find it, sometimes you don't. So there was conflicting reports too. People had very strong opinions about it. And to jump to the conclusions of the second talk is we will compute what the absolutely perfect best arrangement is that is possible for a complex and a real valued field to be aligned to one another if both have the same spatial frequency. Because that is a constraint optimization problem that you can't perfectly solve. But using this language of weakly nonlinear analysis, we can compute what these patterns are like. Then we will actually find that animals produce these specific patterns in their brains. So this weak anticorrelation isn't a failure of measurement. It is very close to the absolute optimum that is mathematically possible under the constraints of a typical spatial scale and an anticorrelation. Right. So now let's look at the details of how we derived that statement. Yeah, for the motivation, this is a cartoon of the tree of modern primates, or at least the primates that ended up in our data set here. Tree shrews as an outgroup, and then the galago, the owl monkey, and the macaque. The little circles here are their color vision abilities. So the macaque is a trichromate. They can see color just as well as we can. Actually, a little bit better, because color blindness is relatively rare among macaques. The galago and owl monkey are both night-dwelling creatures, only 1 cone receptor. They're truly colorblind. And the tree shrew can see 2 different colors with 2 cone receptors. The tree shrew in that sense is very similar to most other mammals out there. Cats and dogs are also colorblind with 2 different cone receptors. We have lost most of our color seeing abilities probably around the age of the dinosaurs, when all our ancestors were these tiny dark-dwelling animals, and there just wasn't a need for complex color vision. Most other animals that are alive today actually enjoy a much more sophisticated spectral sense than we do. So reptiles and birds, for example, have 4-cone receptors. So it's very likely that dinosaurs had that too. We just lost it because it didn't matter too much. We had vastly enlarged olfactory bulbs to sniff our way through dark holes for a really long time. And we could only diversify again after the extinction event made space for all of these new and freely available niches. Here in this tree of life, I've added little numbers, and they show key events in how these primates diversified. So based on the existence of cytochrome oxidase blobs in these 3 species, but not the tree shrew, we can hypothesize that they might have emerged relatively early on in these 3 groups of primates. Orientation domains, number 2, very likely evolved in these brains once they became sufficiently large in tree shrews and primates. But again, our earliest ancestors were so small that it's unlikely to imagine them with a complex system of orientation domains in their brains. 3 is a pretty trippy event because some of the primates returned back into a nocturnal lifestyle after transitioning to a diurnal lifestyle. So there was a branch of primates who lived during the day mostly, but then became dark dwelling again a little bit later. That's the owl monkey here. Then there is the split between the Old World and the New World primates when South America and Africa split, leading to essentially independent evolution of both of these groups of primates. And then here for the macaque, we have the opsin gene duplication that gives rise to trichromacy in the Old World primates, and we are part of that group. Poor owl monkey returned back into nocturnality at some point, and then lost its opsin so that it is truly colorblind. PETM is a thermal maximum that happened around 50 million years ago that led to an extreme diversification of various lineages of primates, and KPG is the extinction event that killed off the dinosaurs. So a lot of stuff has happened over the last 35 million years. There's a good chance that maybe some of them were sufficiently challenging to lead to deviations of the common design and the pinwheel statistics that we can pull out of the circuits that these mammals have in their brains. Okay, so how do we couple cytochrome oxidase and orientation domains together? I had alluded to that a little bit already in the first half of the talk. We can write down a joint model where the total energy of the layout is a free field for orientation domains. That's the common design. We talked about the ECPs, all that stuff. Then we have a second free field for a real valued field. This would be cytochrome oxidase, and some sort of interaction kernel that couples both together. Now for the choice of interaction kernels, well, let's talk about cytochrome oxidase first. This is a relatively easy field. We just want one that produces a spatial scale. So we use our old friend, the Swift-Hornberg operator again, and we want a typical amplitude. So we want a knob with which we can turn how much CO expression there is. Is it a lot or is it very little? And that's going to be it. And then we want to couple them with one another based on these previous reports of an anticorrelation. So we choose an interaction energy where we take the absolute value of Z. So again, this is the orientation selectivity. It's the absolute value, not the phase. The phase doesn't matter for this kind of stuff. It's really just the strength of tuning. And C is the amount of cytochrome oxidase. So if we want to minimize the energy, we want to make this guy as small as possible. And that is small whenever there is this trade-off that you have large orientation selectivity together with very little CO expression or vice versa. And kappa is a coupling constant that tweaks how much these two fields can chat with one another. If kappa is 0, then we have just independent development of both. If kappa is very large, then these fields are strongly interdependent. Right. And later, here's a little cartoon of what the solutions are going to look like. We'll derive those both numerically and analytically. Okay, so the first experiment that we could do is just qualitatively, if this coupling was sufficiently strong, then it is possible that the presence of an orientation unselective bias would change the statistical property of the pinwheel arrangement itself. That's relatively easy to measure. For that, we would only look at orientation domains, do our previous business of counting pinwheels, measuring typical scales, and counting their numbers. if we go through that exercise, we find that the owl monkey, Aotus, and macaques, and the tree shoe all have the same pinwheel density of essentially 3.14. Yeah, statistically indistinguishable. And that suggests that the influence of cytochrome oxidase on orientation preference must be pretty small. Otherwise, you would measure some deviations here. And so we can simplify our model a little bit in that we can modify our optimization problem where we still have the free field of cytochrome oxidase and the coupling, but we take the Z's, the orientation maps, as the optima that we already know, the ECPs. So we now have these ECPs as a bias term on a secondary pattern-forming system, the cytochrome oxidase map, that now has to arrange itself, trying to optimize that energy function. So that makes it an interesting constraint optimization problem. Okay, so how do we go about it, and what does this EC look like for cytochrome oxidase? This is the same style in which we wrote the energy optimization problem for the complex field earlier. It's 4 terms. The first one with this LC, that's the Swift-Holmberg operator, that is the term that produces the typical scale, just like we had seen with the complex field. Then we have a quadratic and quartic interaction. They make sure that our expected value doesn't vanish, kind of like the Higgs mechanism. We have a finite expected value. And then we have a term that breaks the symmetry. And with that, we can have either a bias towards a lot of expression of CO or don't break the symmetry. And that will change the solutions of this free CO field. The coupling, again, is just multiplicative, where we make an interaction term between the absolute value of the complex field, the tuning strength, and the amount of cytochrome oxidase expression. Okay. So then we can compute our dynamical equations. Again, this is variational. We want to find the energy optima of this guy, so we do gradient descent. We compute the derivatives of this guy with some sort of coefficient that is just how strong this is, and we have our coupling term here. Oh, and this should be a Z, not a psi. That is actually the same thing here. I just took it from different manuscript. Okay, so what are the numerics like, and can we get a sense of the solutions of these coupled optimization problems? This is very similar to what we had seen in part 1. The free CO field will form stripes or hexagons depending on this bias term delta. You can show that with weakly nonlinear analysis. And the couple leads to, so if kappa is finite, then the coupling will lead to coupled amplitude equations. So if we write down our real valued cytochrome oxidase field, now note it has terms on both sides of the critical circle, so that these are real valued solutions. Then we get dynamical equations for how these amplitudes change over time that involve the amplitudes of the cytochrome oxidase field, the B's, but also the amplitudes of the ECP, of the orientation selective field with this coupling coefficient. So if kappa is 0, then we just have a free CO field that doesn't know anything about orientation selectivity. If kappa is larger than 0, then these two fields start to chat with one another. And we can write down the amplitude equations and identify the fixed points of this dynamics where they're exactly 0. You can see the interactions here are relatively complicated because the change of this amplitude J depends on the presence of amplitudes in the ECP at 2 different spots. So either it's away towards the left on the critical circle with minus nu and minus 2 nu, or on the right with plus nu and plus 2 nu. So in other words, the CO mode is active and stable within this enforced pattern from the essentially complex plan form. If J minus nu and J minus 2 nu, that is to the left, are active, or both on the right are active. So that sounds pretty trippy. We'll see later in the numerics how that unfolds in practice. Okay, so advanced numerics isn't really advanced. It's just what we did previously. The only thing is we add an additional bias term. So in our MATLAB code, we'll go to that in a couple, or like 2 minutes, now has this extra chunk where we compute an ECP, which you already know are relatively good solutions to the standard model. We compute the absolute value squared. And this becomes an additional bias term in the integration of the cytochrome oxidase field. So if kappa is 0, then we just have these independent terms. And if kappa is finite, then this starts to matter for the dynamics of the CO field. Okay, so first let's look at the dynamics of the free CO field for kappa is 0. I claimed that it would make stripes and hexagons. I can show you the, this should have been a video. I can show you the video though, because I simulated that on the flight. Okay, so this is the SEO field with a small value for symmetry breaking, and that is going to produce stripes. The initial dynamics are the same. You start with a random pattern, you select modes close to the critical circle, then as time unfolds, most of them die away, and you end up with a stripy pattern. You can see it's still a little bit distorted, but if you were to let this run really, really long, the stripes would become straight. Okay, and if we increase the bias term, the initial part of the dynamics look exactly the same, selection on the critical circle, but then the modes start to rearrange themselves into a hexagonal pattern. So this is the free CO field. And now based on these two possibilities, having this bias term from the orientation map will change these layouts in particular ways, and we can track what this change is like. Right. I had claimed earlier that based on the considerations from the amplitude equations, there's a relatively complicated coupling between which modes are occupied in the ECPs and which modes are occupied in the cytochrome oxidase field. As an example, if we were to start with this ECP here, you can see, I hope you can see the peaks in the spectrum. This is just a typical ECP. Then we have to do this kind of geometry thing and figure out whether these triplets of 3 are unoccupied to figure out which CO modes can become active. That is kind of this complicated interaction on the critical circle. So based on this ECP, we would expect that the following CO modes are active and these are inactive. And then we can simulate this numerically. I'll let this run in a second too. You're already pretty familiar with this by now. Initially, you select stuff on the critical circle. Then the modes start to organize themselves in a particular way, and all the way towards the end, you can see that, I cannot see it that well, but you converge on a solution that is essentially the same what we had expected from the analytics. So there are very complicated interactions between the modes that stabilize these types of CO layouts. Let's run this in the background while I continue chatting. If you want to play with this, this is the code run coupled maps. The two important parameters, kappa again is the coupling. You can make this, let's make this 1. And gamma is the amount of symmetry breaking. So if gamma is 0.5, the free CO field would make stripes. If gamma is 2.5 or larger, then the free CO field would make hexagons. So we can start with stripes, doesn't really matter. And down here is the integration. And that's where I just took a picture for the slides. You can see our old integration with this implicit scheme plus the 3 terms. And one of them is the coupling. If do plot is for, wait. Oh, there it is. Oh, I don't think I can have PowerPoint and MATLAB open parallel. Then let's do it in the break. Right. Or you run the code yourself on your laptops if you want to. Okay, so this is how the orientation map will enforce a certain pattern for the CO layout. We can compute the optimal arrangement too. So what is the best possible way for these patterns to align based on the fixed points of this dynamics? And it will depend on the number of modes in the ECP. That's not too surprising. if we have more and more numbers of modes on the ECPs, we get like slightly different solutions for the CO pattern. But we already know from the pinwheel considerations that the solutions that produce the statistics consistent with what we find in the data, we would need a certain number of modes in the ECPs, otherwise it wouldn't work. Yeah, that's the mathematical term. So we are restricted to a certain range in this parameter space where the pinwheel density is consistent with the 3.4 that we had seen in the data. And that means now that our correlation coefficient is bounded. We can't do better than around 0.3 or thereabouts. Otherwise, we would observe deviations in the pinwheel density. And again, the underlying reason is that we have a real valued field and a complex field where we try to make the real valued field as similar as possible to the absolute value squared of the complex field under the constraint that both have the same typical scale. And that is a thing you can't solve at the same time. But you can go through the math and find the best possible arrangement, which turns out to be at the correlation coefficient around 0.3. Okay, so we can now study this numerically. These are different experiments for different coefficients that as a free field would either produce stripes or hexagons, different values for kappa, so how strongly these fields interact with one another. And you can see here for the observed arrangement of CO orientation maps, we observe the optimum of around minus 0.25. And if we displace CO from the orientation map, just move them away from one another, then this arrangement goes away. This is why these curves start to go up. That's the control here. So we optimize both of these fields at the same time, and then we look at displacements. So that seems to confirm our analytical insight. This here is the phase space of our model. If we have very small coupling strength, in some limit it becomes irrelevant that it exists, then we recover these two possibilities for the free CO field. We either get stripes or we get hexagons. And when we crank up the coupling, the kappa, at some point, irrespective of whether it's CO bands or hexagons, we transition into a state of irregular CO patches that are not perfect stripes anymore, or hexagons, that inherit some of the structure of this pinwheel layout by trying to move these CO patches so that they align relatively well with regions of low orientation selectivity under the constraint that there's a typical scale. So they'll start to move around a little bit. I can show you how this arrangement works. So if we take a particular cut through our solution space, so say we start down here with perfect bands, and we slowly increase kappa asymptotically, and just look at the fixed points of the dynamics. We observe the following dynamics. So this plot is a little bit complicated. It shows the ECP in the background, the orientation domains, and the stripes are the cytochrome oxidase pattern, but it's just contours so that you can see both. So these vertical lines just means that the CO pattern is stripy. And it's stripy because we started at kappa equals 0, so somewhere down here. And then we increase kappa slowly, and for a little while there's not much happening, but once we are in this regime of maybe a third or so, we start to notice that the stripes get distorted. So the stripes start to arrange themselves a little bit around the underlying patch of the orientation map. That means we are now in this regime here, where we lose the bandedness and transition into something new. And once the coupling is sufficiently strong, it's essentially like a switch where the CO field finds the optimal arrangement between both, right around here. Whoops. Can I stop this? Yeah, right around here. And now you can see that these CO-rich regions are relatively disorganized. They're all over, they're irregular, but they align relatively well with minima of orientation selectivity. So here's a ridge with relatively low orientation selectivity. Notice the darker colors, and it sits in this region of higher CO expression. And that is the best that one can do under the constraint that both have the same typical scale. The complementary video is over here, where we start with hexagons in the absence of coupling. So hexagons, if you draw a contour line, you just see the peaks. So you see kind of the hexagonal array of rings. And if we increase this coupling coefficient slowly, the first thing we observe is they start to deform. And this happens at slightly larger values in comparison to our earlier simulation. No. Around 0.6 or something, or 0.7, where these distractions become noticeable. So now we are entering this regime up here. But then once the coupling is strong enough, it also just flips into essentially the same pattern of CO bands that we got in the stripy regime. So that means that in the limit of large kappa, the free dynamics of the CO field are essentially irrelevant because it is completely governed by the structure enforced from the orientation selectivity domains. Whereas when the coupling is weaker, we see a little bit of an effect of both. And that is tuned by this kappa coefficient. Once we have these arrangements, we can look at what they look like by computing, this 3-point correlation between both and their spatial arrangements. So we can shift, say, the orientation layout relative to the CO layout a little bit and measure how this correlation falls off with space. If the arrangement of these maps is in fact optimal, then we would expect that this 3-point correlation as a function of space would have a minimum for y equals 0, some number that the particular data set managed to create, and then it would just increase as y goes up. And we also know the analytical limit. We can't do much better than this number. That's the best arrangement between these two maps that is possible. In the model, depending on where we are on these choices of kappa, these are the two cartoons that illustrate these 3-point correlation functions. Notice in the center, there's always a minimum. That is the best arrangement that's possible here. If we displace one map from the other, this correlation increases. The arrangement isn't as good anymore. And about 1 lambda away, we find a secondary minimum because these maps are always roughly periodic. So the correlation functions will reflect the same structure of periodicity. And now what's cool is we found, and then in the same parameter space, we can also compute this 3-point correlation function for the different values of coupling strength and CO expression. We find our old regimes, the CO bands, the hexagonal patches, and this irregular regime. And only in the irregular regime is the arrangement as good as it can be, and it approaches asymptotically this theoretical limit of about whatever that number is. Okay, and if we look at the data, then turns out that our data is actually very, very similar to that. These are the same 3-point correlation functions measured between the CO and the orientation domains. And here are the radial averages. So you just integrate out the angle, and you find that the macaque actually is relatively close to an optimum here and seems to approach the theoretical limit. Whereas the Aotus, one of those not dark-dwelling creatures, and the Galago don't seem to approach the theoretical limit quite yet, even though the pattern of them seem very similar. This here is the statistics of the entire data set, and we find again that the macaque is relatively close to the theoretically possible limits of what one can find. So even though this seems, in terms of an effect size, like a relatively small anticorrelation, it is actually the best that you can do under the constraints of the system. And what that tells us is that even though there are these invariants for things like pinwheel density, that the CO blob placement became optimal relative to this invariant layout of pinwheel domains in the macaque. And considering the evolutionary lineage, this must have happened relatively recently, in fact, over the last 35 or 40 million years. And it did not significantly change the layout of pinwheels either. And that is further evidence that there is something truly important about orientation domains and a pinwheel density close to 3.14. Otherwise, there might be more measurable deviations if this coupling really exists. So it took the macaque around 35 million years to rewire its brain to include this layout of cytochrome oxidase-rich blobs without interfering with the layout of orientation domains. Okay, that was the coupled map story that I wanted to chat about today. because I felt it follows somewhat naturally to the math that we had done earlier today. For the last 15 minutes of today, I want to talk about something completely different. And that actually echoes one of the questions that came up earlier about pattern formation in the brain at large. And I want to show you a picture for that, which I thought is absolutely bonkers, was awarded with another Nobel Prize. So how common is pattern formation in the nervous system? These are discharges of a neuron in a particular region of the red brain called the entorhinal cortex. Every red dot is an action potential, so one of those pulses of electrical activity, when the rat is running around a square box. The way you do this experiment is you have the rat running around, you usually throw in little yogurt gums or something that the rats like to eat, and that keeps them running, and you collect data like this for a little while. And when you look at this pattern, that looks like a hexagonal pattern, which is just really remarkable as a tuning property. And if you think about pattern formation a lot, hexagons are just something you typically get. What's also wild about this is grid cells, that's the technical name for these guys, don't just exist with one spatial scale. They usually fall in certain groups. And if you probe deeper and deeper along the entorhinal cortex, you find different grid modules, so neurons that have the same hexagonal tuning profiles, but the spatial frequency is different. Here's a little cartoon from a paper, and that too smells like pattern formation, where you just change the spatial frequency, but you get the same kind of hexagonal layouts. So for some reason, brains seem to do this, and what's cool about grid cells is that spatial navigation or spatial cognition is really complicated behavior. There's some actual brain stuff happening. With the orientation maps, one could always argue, well, maybe this is an idiosyncrasy of sensory processing or perception, or the way edge detection works in highly tweaked circuits. But this is something that animals need to do every time they run around in an arena. When I transitioned from my PhD to my postdoc, I wanted to study representation of knowledge a little bit more and get a deeper understanding of how neural circuits operate when animals are actually engaged in complicated tasks. So to do that, I use a task in virtual reality. So that sounds pretty trippy, but the reason why this is done in virtual reality is pretty simple. If you want to use a big microscope or heavy electrodes, mice can't carry that on their heads. But you can fool the mice into thinking they move around in the real world if you immerse them in a virtual reality. And then you can use all the advanced optics and electronic tools available to get high quality data out of the mouse brains. So the task looks like this. In a simulated corridor, the mouse has to run along the stem of a T-maze, while bright cues appear to the left and the right of the walls. And the mouse has to turn in the direction with more of these cues. And if the mouse does this correctly, it gets a droplet of sugar water. Here's a video of it. And try to play it yourself. What's neat about this task is they have to do two cognitively demanding things. First, they have to solve the maze. They have to know where they are in this world, because that will determine how they have to run, what they have to pay attention to, and so forth. And the second thing that they have to do is count these towers. Well, they probably can't count, but they have to produce a sense of numerosity or accumulate this pulsatile evidence that comes about. And then they have to integrate both of these data streams somehow into a representation that exists in their brains. While mice did this, one can use microscopy to record from large populations of neurons. And this is a little example from two-photon microscopy. So this is fluorescence of GCAM, that's a fluorescent calcium indicator. And this was recorded while mice were actually running this very task. And so one wonders, well, how does the neural activity relate to what these guys do in that maze? Like, can we find echoes of the towers, of where they are in the world, and so forth? One way to analyze data of this type, well, first you process it a little bit and transform this into time series. So now you have a giant matrix of number of neurons, number of nerve cells in your picture, something like 500 or 1,000 times time. And you try to find structure in this data set. Historically, people talked about tuning a lot. So you would take neuron 1 and try to figure out neuron 1 is particularly active when the mouse is 30 centimeters into the maze. Neuron 7 is particularly active when the mouse is at the end of the maze, and so forth. But in recent years, people got really excited to better understand the coordinated activity of neurons as a true population state. And to make analysis like this, you have to record from large numbers of neurons simultaneously. You can't just do sequential experiments where you just record one neuron at a time. When you have data of this type, many neurons over time, you might wonder, well, how rich is this data actually? In other words, what is the dimensionality of this data? Now, what do I mean by that? If you had a data set with 1,000 neurons, conceivably, they could live in a 1,000-dimensional state space, where every axis is the firing rate of a single cell. So that is scary complicated. It turns out that in many different behaviors, and I'll chat about this a little bit more in the future, neural activity doesn't really fill this high-dimensional space. In fact, neural activity is vastly lower dimensional. So we're talking about like 3, 4, 5-dimensional structures, manifolds, that are embedded in these 1,000-dimensional spaces. And if that is so, then the challenge of extracting this data is to find, as you would do for any other manifold, a suitable chart that maps this geometric object so that you can express where you are on this manifold in terms of a smaller numbers of coordinates. The same as you would use longitude and latitude to describe your position on the Earth. But now we want to do this with neural data. Popular for this are nonlinear dimension reduction techniques. I use one that's called manifold inference from neural dynamics. The way this works is you take pairwise firing rate vectors. So here's one I, here's one at time point j, and you compute a similarity of both based on the probability that j would follow I. That gives you a very large pairwise distance matrix. And then you try to compute, and if you zoom in, you already see structure. So these two states are relatively close to one another. So they're very likely close on this manifold. Then you try to find a set of latent variables so that the pairwise distance in a lower dimensional space is as close as possible to the entries in this matrix. You can do this, then the neural trajectory from a 1,000-dimensional space becomes a neural trajectory in a, for example, 3-dimensional space where we can plot it. And we can color code certain features, for example, where the red was in the maze. And this is shown here. And what you can visually appreciate is that position in the maze forms a smooth gradient on this neural trajectory. And this is something the algorithm didn't know. The algorithm only tried to find structure in neural activity. It didn't know that this data set came from a rat exploring a square box. But the algorithm found a chart for this low-dimensional population state where position in this box is represented as a smooth gradient on it. Okay, so this is just one example. It's also the case in the towers task. And to show you that this works relatively well, we can take about 10% of the neural data that we had recorded, 40 neurons out around 400. We can then project this activity down to a handful of latent variables and reconstruct the data. And visually, you can already appreciate that this is super similar. So even though we have so many neurons in the data set, the actually underlying population state, their covariance structure, is much, much simpler. It's a much lower dimensional state. You can try to estimate the dimensionality too by changing the size of this bottleneck. So you constrain your manifold that you fit to be 2-dimensional, or 3, or 4, or 5, and you see that it saturates at a certain point around 5-ish or so. So there's a relatively low-dimensional manifold that analyzes the behavior. Now you might wonder, well, that is pretty data-heavy, but how can that possibly be true? Like, how can you describe the activity of 1,000 cells, not averaged or anything, but as a continuous time series with only 5 numbers? And the underlying mechanism that translates dynamics on this manifold to neural activity is something that people sometimes describe as firing fields. So here you see an example of 25 different neurons. Their firing rate is color-coded on the same low-dimensional manifold. And you see they form these hotspots. And that means if your population state is in a particular state on this low-dimensional manifold, all the neurons whose firing fields overlap in these regions become active at the same time. This produces covariance, and this covariance is what reflects the low-dimensional population state. So these cells tile the low-dimensional manifold. And the conceptual picture now is that we have these firing fields on a low-dimensional manifold that might represent behaviorally relevant variables. And we had seen position already. But if we plot position and accumulated evidence on the same manifold, so notice that this is the same object here. You can see the 3 dots up here. we find smooth gradients of position and evidence on the same low-dimensional object. So that means even though there's like 1,000 neurons that are coactive at the same point in time, the activity can be represented by a handful of variables in a latent space. And in that latent space, key behavior variables, for this task, position and accumulated visual evidence, are represented by smooth gradients. And I also brought you a video from the full trial. Oh yeah, and these are 2 trials in this latent space. They start at similar position at small, at the beginning of the T, and then they kind of diverge as the evidence becomes either large positive or large negative. I also brought a few videos to embed the complete time series without any chopping. So what I mean by that is the actual experiment is about an hour and a half long. So what you do is you have your 500,000 neurons times 90 minutes with a relatively high sampling rate. And you can give the entire data set as is to one of those dimension reduction algorithms and ask it, find the latent geometry of this data set. The algorithm does not know anything about the behavior. It doesn't know that there's position involved or accumulated evidence. It doesn't even know that's from a mouse. It's really just trying to find structure in this giant matrix. So here I have done that. And I will plot the same manifold with 3 different variables: position in the maze, accumulated evidence, and whether the mouse made a correct choice or false choice. And let's just go through these individual examples. This is position in the maze. You can see it's a really complicated structure, but you can start to interpret it. So the mouse starts at low values in the beginning of the maze and then moves forward towards the T. And then when it is at the end of the T, made a left or right choice, and the neural states start to flow back to the beginning of the maze. Because the trials follow one another in time. There has to be some sort of transition between them. If we plot accumulated evidence on the same neural manifold, we find that orthogonal to this position gradient, it is now split into 2. You can see there's blue on one side and yellow on the other. So there is a mental representation of whether the mouse has seen more evidence on the left or more evidence on the right, the kind of variable you would need for decision making. And the last thing that's a little bit obscure is why are there 2 paths that go back to the origin? And if we plot whether a trial was correct or an error trial, we see that this splits these 2 branches that go back to the origin. While the mouse is running in the T, it doesn't know whether the trial is correct or not. But at the end, it either gets a droplet of sugar water for correct trial or it doesn't. And that changes neural dynamics in their brains fundamentally before they can start the new trial. And so you build these topologically really complicated objects that reflect the problem of the task at hand. And somehow the mouse's brain has just done that. Like in practice, the mouse has encountered a problem and it has done this problem a couple hundred times to figure out how to get the droplet of sugar water. And somehow neural circuits in the mouse brain have changed to produce these super low dimensional structures in the neural activity of these thousands of cells. And in that sense, there's a certain similarity to what we talked about earlier today. These visual cortical neurons, there are also millions of them. So conceivably, they could also live in a million-dimensional state space. But for some reason, they built orientation preference maps that are strikingly lower dimensional. There's an enormous covariance between what these neurons do that reduces these dimensionalities, and we can start to build effective models on this effective map of the lower dimensional structure. With orientation map, I've shown you an example earlier today. With these representations of knowledge, we aren't quite there yet. But what I'm trying to communicate is that the flavor is similar. This is just a common property of neural circuits. Right, we saw the videos. I'm not the only person to make this argument. I mentioned it briefly earlier too. If we look back over time, the number of simultaneously recorded neurons has grown exponentially for many, many years now. Here are examples from my own work, calcium imaging or neuropixel probes. And even though we record regularly from many, many neurons, it has turned out that in the state space perspective, where neurons represent an axis and activity is a trajectory over time, over many behaviors, neural activity lies on structured low-dimensional subspaces. And these structured subspaces seem to reflect the tasks that the animals are executing. And to me, this is a super exciting bridge to make it possible to develop effective theories how the brain operates. If we really had to describe the activity of every single neurons, oh yeah, and this is, more and more people are getting excited in neural manifolds. If we really had to describe every single neuron individually, it's almost like hope would be lost. Like, how would we ever come up with a computational scheme to interpret that? But on this low dimensional level, there is actually hope. An analogy to that might be the Hodgkin-Huxley model. That has demonstrated that we don't need to model every single ion channel in the cellular membrane of a neuron to make sense of the action potential. And I think something similar is happening here. Now that we can regularly record from so many neurons at the same time in complex behaviors, we see that there are much lower dimensional effective processes that unfold in neural circuits. And it is that level of abstraction where systems neuroscience and computation and maybe even psychology can be linked with one another to bring these two domains together. Okay, that brings me to the summary for today. In the first talk, we chatted about orientation selectivity and the pinwheel density close to 3.14. We talked about the theory of orientation domains. Then we chatted a little bit about representations of knowledge. That's my ongoing work. And I try to communicate that I think low dimensionality of neural activity is a common theme, and that this will be one of the key drivers of future insights into how the brain operates. Yeah, so we can chat about this a little bit more. But before we do that, I want to give you just a brief glimpse of my actual work that I do in my lab. I'm still interested very much in sensory and cognitive processes, and historically they have been considered separated. So what I do now is we record from large numbers of neurons in the visual cortex while animals solve complicated tasks. And we find the same low-dimensional manifolds there. And then it becomes interesting, what are the rules, how are they similar, how are they different, which effective theories describe them. And the principal technology for that is two-photon microscopy. This is a microscope that I've just designed for recording while mice behave in virtual reality. I've pasted the picture here to give you a sense of what the experiments are actually like. So the mouse is running on a styrofoam ball. So this white round thing is a styrofoam ball that floats in a cushion of air. And the mouse is head fixed by these two holders. So if the mouse runs forward, what it's actually doing isn't really running forward because it's head fixed, but it is pushing the styrofoam ball backwards. And because it floats in a cushion of air, there's almost no resistance. And we can monitor the change of movement of the styrofoam ball to compute projection patterns that on a screen that surrounds the mouse. And while it is doing that, we can use two-photon microscopy to record from large numbers of neurons in these animals. Rightio, so that is ongoing work, and hopefully it will lead to some deeper insights about how perception works through this lens of low-dimensional population states. Yeah, if you find this interesting and you don't do a PhD yet, consider coming to Delaware. Yeah, acknowledgements. For part one, again, like much of this is the brainchild of Fred Wolf, so he should be mentioned here first. And then Wolfgang, Joscha, and Matthias are the folks who have spent many years with talking about orientation maps. For my more recent work, I was a postdoc with David Tank and Carlos Brody in Princeton, and I've had the pleasure to work with these fantastic people over the last couple of years before I moved to Delaware. And with that, thank you for your attention, and we can take questions. For that, I will go back to my summary slide.
Thank you, Manuel. So, questions? I have actually somehow a tricky question, maybe. So, if the representation in the brain is really low dimensional, so why do we have so many neurons? So, what's the reason that if you only need maybe just a low level representation?
I have a hypothesis.
Please.
Okay, but I have to wing it. I try to write it up, but it's kind of one of those things that you think about in a shower. So what I think is happening, picture you want to represent a 10-dimensional manifold. Like, that doesn't sound crazy complicated, right? So if you have a 10-dimensional object that lives in a very high-dimensional neural activity space. So these are positive numbers, and there's about 1,000 of them. for our neurons, or maybe a few million if you take the whole brain, and then there's this structured manifold in it that is lower dimensional, and maybe it's around 10. Let's take that for the sake of argument. D around 10. Okay, so for a 10-dimensional manifold, what kind of resolution would we want to get to represent a point in a 10-dimensional manifold? So say we want to represent every axis with 10% precision. So we would have 10 states for every single axis to the power of the number of dimensions that we want to represent. So if you do that, like 10 to the 10 is 10 billion, is a relatively large number of states already. So how do you represent that many states with unique activations? Like the simplest scheme you could come up with is state 1 is neuron 1, state 2 is neuron 2, state 3 is neuron 3, and so forth. And so you could try to link the number of neurons with the dimensionality by saying this thing has to be roughly the number of neurons that we have available to uniformly sample this d-dimensional manifold.
I see. So if I understood correct, the representation is low dimension, but the possibilities are huge. So essentially you have to have that many to have all the possibilities. That's like, I see. It makes sense. Okay.
Yeah, I think this is it. To me, it also makes intuitive sense. Like picture yourself playing a game of chess or something, right? When you think about that, chess is also crazy complicated, but when you think about moves ahead, you don't really consider 10 million moves. You have a very low dimensional understanding of the game of chess. You only explore a certain subset of moves that might be relevant in the future. And the thing that people can play chess without even looking at a board, just in their minds, I think that is possible too, because they don't really keep all 64 fields in their heads, but they project it in a much lower dimensional space because they're so familiar with playing chess, so that they can reason in these abstract spaces. So the problem is figuring out what the geometry of these low dimensional manifolds are so that they align with the task at hand. Or a different example might be music. If you play certain chord progressions, that is really hard to remember too, but once you've played enough music, you get a sense of what is like common, what happens again and again and again, and you start to see these structures. And then these mental representations change and align with what has to be done. And to me, that is the most exciting part of cognition too, how we find these structures in the world around us that can be so effective despite their relatively low dimensionality. Yeah.
Thank you.
Yeah.
So when you show the figure of the three-dimensional latent space, there seem to be like two holes, right?
Mm-hmm.
So why is that? Is it because
so let's do it with two colors. We have blue and we have red. Okay, so if the brain state starts here. And the first possibility is the mouse is running forward, and then there's more and more towers appearing on the right side. So the mouse is committing to a right choice trial. I'll do that in orange. Or there are also trials where it's just going to be a left choice trial. And then it's not the real number of towers, it's kind of the internal estimate of the mouse. So sometimes that's correct. Actually, frequently it's correct because they perform this task well, but it's not always correct. So sometimes the mouse takes a left turn here, but then it doesn't get its sugar water. So the mouse here is in a mental state of, oh, I screwed this up. This isn't correct. And yet it has to go back to the beginning for the next trial. And that produces a particular trajectory. Let's do purple for the error trials. And then there's also one for these error trials. But then left and right can also be correct trials, where the mouse got the droplet of sugar water, and those will be structurally different because the mouse has to do different things. First of all, the mouse is probably happy with itself that it got the droplet of sugar water, but later down the road, it will also have to do things like licking, for example, or maybe cleaning its whiskers when they got all sticky from the sugar water. So the neurodynamics in the brain become different. And this is how this topology emerges in this relatively high dimensional space. That's also, to your question, these manifolds can have topologically really complicated properties, depending on how the task is structured. Like, God knows how the chess manifold would look like. There might also be some shortcuts on it or some holes. It's very, very difficult to predict that initially. But again, I think that this is the most exciting future research avenue for computational neuroscience, because we know so little about how these mental representations emerge when we try to make sense of a complex new problem that you have never seen before. Like for a mouse, this is already pretty hard. It takes them a couple of weeks to learn that. And I think the analog to that would be something like chess or playing an instrument for a person.
Thank you again, Manuel, for the lecture. I have another question in the sense of you related your pattern formation on the brain, or orientational pattern formation, to collect motion, to collect behavior of the neurons. And my first question is that if you connected this behavior as a collective effect of the neurons, another question is, you observed topological defects in this. You mentioned it, but what kind of topological defect you observe?
Yeah. So for the first question, the representation of like these guys, these manifolds that I chatted about, at least in the examples that I have studied in the hippocampus, that doesn't look like pattern formation. There wasn't anything hexagonal about them. So pattern formation only seems to hold for entorhinal cortex, these grid cells, and for the visual system. And then topological defects, that is because when you have a pinwheel, so like this point, and neurons on this side prefer this orientation, neurons on this side prefer this orientation, and then you can fill in the others. If you integrate a contour around the defect, you're left with a number. And it's either plus or minus 1/2, depending on whether these orientations change in a clockwise or an anti-clockwise. The orientation moves smoothly around it, right? But there is a singularity in the middle for the pinwheels, and there's also an intuitive reason why it's a topological defect. The picture you had a layout of orientation domains, and you would just smooth it. These roots wouldn't go away. They might move around a little bit, but you can't really do anything to make this disappear, because there will always be a point where horizontal and vertical just clash at one another. And you can start to move it around, but the only way how you can remove pinwheels is you find one with the opposite sign, like the opposite topological sense, and they can annihilate. So they can come closer and closer together, and then they disappear. And actually, if you integrate the contour around both of these pinwheels, you would get a 0, because there's no net charge left in the bounded domain. It's the same as with electrical charges. Like you integrate over the surfaces, and you get a number of the amount of charges in the volume. And it's the same business with the pinwheel.
Hello. So when you showed the 3 manifolds, it was different measures of the same task.
It was actually the same session, same task. The only difference is the color code on the 3 manifolds.
Okay.
So every point is a point in time. Like every single blue dot here is a point in time, projected from the 1,000 or so neurons to this 3-dimensional embedding. You can see like this pattern of these little dots is exactly the same. The only thing that I did different for the 3 is I color-coded the dots here with position in the maze. So every dot is now where the mouse actually was at that point in time. Here is what kind of evidence was visible on the screen. And here is whether the current trial that the mouse is in was correct or not.
Okay, so this idea of manifolds is new for me. So we should think that there is one for each task, like different manifolds would exist in different tasks.
I think there are. I haven't tested myself, but in the neuroscience world, there's this idea of remapping. So you can have place cells that respond to position in a box. But if you give the box a different color, so you make it, I don't know, from green to red or something, or change the texture on the walls, the place cells change. Like a neuron that was a place cell in box 1 might not be a neuron in a place cell in box 2. And that feels like there's now a new low-dimensional manifold that represents this environment too, that is relatively unrelated to the representation of environment 1. So all of these guys seem to exist in the shared neural space. So there might be a second manifold that has some different dimensionality that coexists.
So related to his question, this is also like if you need a lot of manifolds, so you would also need a lot of
space, yeah.
a lot of neurons.
People have done some very cool statistical physics work on that. So if you have like random, not random dots, but random manifolds, so groups of points that have certain properties in high-dimensional spaces, how much space do you need to make sure that they're always linearly separable, for example? That is something you can just compute. If you don't have enough neurons, then these manifolds would start to overlap. And so you can do some exciting physics with that. It all comes from the work of spin glasses. There's some recent work from Heimson Polinsky where they do a lot of the statistical physics of neural manifolds, and that is linked to that. Like how many manifolds can I fit into a n-dimensional space, and how are they related to one another? Do they overlap? Do they intersect, and so forth?
Thank you.
This idea of our representations of knowledge of a task in a manifold is very interesting, but I was thinking, when we think about the complexity of human cognition in relation to other animals, this could be just our capacity to represent more complex, higher dimensional manifolds, or there will be some other difference?
Well, I don't know. My hunch is it is. And my second hunch is a logarithmic thing. So even though we might have 10 times as many neurons, we can't mentally imagine 10 times more dimensions. I think the increase of our cognitive abilities over a mouse are relatively incremental relative to a mouse to some simpler organism. I don't think a mouse could learn chess, but it's like the distance seems to be smaller than to other organisms. But I don't have much evidence to support this. It would be super cool to train mice on really cognitively demanding things, like make them count prime numbers or something, but I don't know how to train a mouse on that. They're good with tasks like that, where you give them sugar water depending on a complex sequence of choices, but or at least I don't know how to study a behavior that actually requires inference or compositionality or like building complexity out of simpler elements, like we learn stuff. Like when you learn about Fourier transformation for the first time, or for me too, I remember that was extremely confusing. Like I didn't get it at all. All these complicated exponentials, and you integrate it, and it didn't make any sense. But after a while, you just get used to this style of thinking. And then Fourier transformation is just another word that you can use to form more complex thoughts. So when we reason, there is this compositional nature to it, that we can build from simple elements more complex stuff. I think math is one of the greatest examples for this. And if I only knew how this would be possible to study in mice, I think this is what makes the human reasoning abilities so remarkable, that we can learn something that's hard, but then it becomes a thing, and we can start to reason with that further and further and further to build into infinity.
Yes, makes sense. Like we compose these manifolds, creating a more complex structure. Yes, it's a very good answer. Thank you.
Thank you. Very interesting work. I was wondering if, if the neural manifold somehow represents the network of neurons and the network topology, would you think that during learning, as synaptic weights change, the manifold itself would change? And would you think it becomes more or less complex with learning?
We've measured that. So during that task, mice can't just solve this T-maze. So the way this actually works when you do that is it's kind of like Mario Kart. The mice have to do level 1, which means just run straight for a meter. Then you get your sugar water. And once they do really well, they have to learn how the T works. So now you have to turn left and turn right. There's an alternation task. We go left, right, left, right, left, right. And then you start to introduce these cues that give the mice a hint of where their reward is. And there is one stage in the shaping procedure that we call the visually guided task, where in the visual reality you see this giant marker. It's like a sign that says, here's your sugar water. You gotta run here. But everything else in the world is already the same as in the final maze, the towers and everything. but they can still use the marker to figure out where their sugar water is. And you can do the same kind of analysis that I've chatted about earlier, record from many neurons, try to estimate the intrinsic dimensionality of this manifold, and it turns out that that manifold is lower dimensional. So it seems that during these earlier mazes, when they don't have to accumulate evidence yet, but they need to know where they are in the maze and all this kind of stuff. They do build mental representations that are already tailored to the task, but as they start to learn new things, the manifold seems to adjust. And not just in a topological sense, but in this case, even the dimensionality increases by this one number that is very suggestive of this just being evidence as a new variable that they didn't need to know in the earlier versions of the maze. Yeah.
So going back a little bit with the pinwheel. So do you have any conjecture why pinwheel in the end?
Oh, no.
No. Any speculative idea?
So we played with a few ideas during, should mention Michael Schnabel here, who had the idea that pinwheels might be linked to what they thought are intrinsic contours in the world. So I usually color code orientation preference in these HSV color maps that you have all seen a million times. A different way of representing them is if you have your two-dimensional cortical surface, you have little like bars about what kind of orientation these different neurons prefer. And sometimes you have a pinwheel when two neighboring orientations are almost orthogonal, but typically they will smoothly vary until you have maybe another pinwheel. And what you can do now is you can connect neighboring neurons following these intrinsic lines, like that. So you can compute a line so that the gradient of this guy is the preferred orientation. So these lines have the property that they would elicit the largest possible response for these kinds of layouts. And the hypothesis, and I think they're still working on this, is that when you look at the ensembles of these lines that are produced by ECPs on the brain, that you find that these contours are particularly important to make sense of natural scenes. So that there's a link to natural scene statistics by just looking at the properties of edges and the orientation of edges. Yeah, but that's a hypothesis. I don't think anybody has conclusively demonstrated it, and we don't know how to change in animals. If we had an experimental method of making a cat that doesn't have orientation domains in its brain, that would be the best test. But nobody has figured out how to selectively manipulate these circuits in that way without disrupting vision itself.
Thank you.
There's one more.
About the reduction of dimensionality, those are not related with common patterns in the brain. For example, some patterns, for example, form when you are static.
Mm-hmm.
And yeah, my question goes in the...
Yeah, so they are not obviously related with things like LFPs or EEG signals or MEG signals. Like other people look into like these collective phenomena with electrodes and you look at average activity. I haven't explored that systematically. That said, I believe the reason that you can decode informative things from these super average properties like EEGs and LFPs and MEGs, fMRI too, is because the underlying neural population code is so low dimensional. Like if all of these neurons were truly independent and then average like 1,000 of them would be borderline impossible to decode relative to the task at hand. But yet our colleagues who work with EEGs, even in human subjects, can decode quite remarkable things from these super average time series. And I think that it's kind of like a renormalization thing. Like you have some complicated microscopic state and you start to average out certain degrees of freedom, and you're still left over with something that's sufficiently informative to make an informed guess about what the collective state is like, even though you have not observed every single neuron that went into this process. Right. So I can't make any specific statements about how this is linked to like theta oscillations or alpha waves and things like that. But it would not surprise me if there was a deep relationship between the low-dimensional cognitive manifolds that we see here and these phenomena that people have explored for many, many years now. Yeah. Did that answer your question?
Let me ask if that answers your question.
Okay.
Okay, let's thank Manuel again. Thank you.
My pleasure.
Okay, so I think so we end our morning session now, and at 2 there will be another talk by Manuel for the entire institute. So please just arrive on time. Okay, thank you.